Welcome, everybody. It's lovely to see you all for our monthly CDCR seminar. I am really, really pleased that we have a most wonderful guest this month. We've already had a wonderful visit, and many of you have already had a wonderful visit with Dr. André Veillette. He, of course, is a world-renowned immunologist and medical oncologist, and he has always been interested in signal transduction and mechanisms in the immune system. So over the past 3 decades, Dr. Veillette has really characterized numerous intracellular molecules and receptors that play a very crucial role in normal immune regulation, but a number of these molecules have also been directly or indirectly implicated in the pathogenesis of leukemias, lymphomas, and other cancers. So these molecules have been linked to the development of immunodeficiencies and autoimmunity, so very widespread impacts, and just shows the power of seeing everything through an immunologist's lens. These proteins are all potential targets for therapeutic immunoregulation and the treatment of cancer, autoimmune diseases, as well as viral infections. So Dr. Veillette has always used state-of-the-art genetic, molecular, biochemical, and cellular approaches to study these molecules of interest. And of course, his work has resulted in very high impact and leading publications. And he's really set the bar high for the rest of us. He's obviously won so many awards and prestigious grants that they're too numerous for me to list. And of course, has been invited to conferences all over the world to organize very important international scientific consortia. He's also on the editorial board of many scientific journals. Most notably, I wanted to let you know that he's recently had, I think, very large impact in his role as the executive director of the Terry Fox Marathon of Hope Cancer Centers Network. And I think it's 54 centers that are involved now. And André has had to coordinate and manage all of the data coming from all of these sources. And you guys have probably seen several recent high impact publications have come out of this. So the work has really been officially delivered. And we know how hard it is to execute on these super large consortia, and things that come out of them often seem very theoretical, but it's wonderful to see how you've just made it all happen, André. So on that note, I want all of you to help me welcome him to speak today about a very exciting news story. I've been waiting all day to hear about this, and I understand it's coming out in a very big journal soon. So help me welcome Dr. Veillette, everybody.
Thank you, Sheila. This is very kind. Coming from, it's always, I think everything she said is also true about her. So thank you, Sheila. So, this Marathon of Hope thing that I've been doing for like 4 years now, I think it's, of course, we worked hard and in getting these 54 centers to work together and all these genomic data. But really what has been the most helpful is the spirit of Terry Fox, who's one of the only modern Canadian hero. And I think he inspired all of us, and he inspired people to get involved. And I think without his vision behind us and his support that the government supports as well, I think we would not have been able to do that. Anyway, so he's there. So as Sheila pointed out, we've been working for a number of years now in figuring out how the immune system is controlled. I used to write grants like 40 years ago, almost now, 35 for sure, saying, oh, if we know better how the immune system is controlled, then we can develop better treatments against cancer. Of course, I was writing that 35 years ago, but I didn't mean any of it. I just wanted to get my grants. But I think what we realized over the years, especially the past 20 years, is that it is actually true. So if we understand better how the immune system works, we can develop better therapies against cancer. And if you put aside the targeted therapies like the kinase inhibitors that are also quite effective, basically the most significant advance in cancer treatment has been based on a better understanding of how the immune system works with the immune checkpoint blockers, right? Or even the therapeutic antibodies work by stimulating the immune system. So I think it's now become a reality, and it sort of encourages me to continue to try to elucidate how the immune system works, but also not only do fundamental research that enable that, but then take that information that I have generated to hopefully orient it more towards the development of these therapies in a more concrete way, and perhaps even developing some drugs that can achieve that. It's hard in Canada, we're talking about this, it's hard then to take it to the clinic and develop these phase 1, phase 2, phase 3 trials. And that's a bit of a problem with the system that we have here, but I'm still trying. So, I think just as a general rule, immune cells are controlled by the balance of stimulation by two different types of receptors and pathways. So you have on the one hand, the activating receptors, and this is true for any kind of immune cell, the activating receptors that promote immune cell development, immune cell activation, and you get the inhibitory receptors that basically prevent that or terminate that. And it's, if you have a preponderance of inhibitory signals over activating signals, you don't get immune cell activation. And in the context of cancer, for example, it can lead to tumor progression. And we know this is true through a variety of different types of observation, including studies of humans that have immunodeficiencies. Now, if, however, and that happens under most physiological circumstances, but can also happen pathologically, if there's more stimulation of activating receptors compared to inhibitory receptors, then you get immune cell activation. It's great to protect us against a variety of pathogens and against cancer, but if it's excessive, it can lead to autoimmune disease and inflammation. So playing with these activating receptors, immune receptors that we call also checkpoints, can potentially influence the activation of immune cells and, of course, control the ability of immune cells to suppress tumor growth or favor tumor growth and protect against autoimmune disease and inflammation or cause autoimmune disease and inflammation. So I'd like to talk about two immune cell types today. So I've already apologized to people who work on NK cells. We used to work on NK cells, then we stopped working on NK cells, and now we're starting again to work on NK cells. And I think you'll understand at the end of the talk why. But we're except for B cells, I don't like B cells, okay? So except for B cells, I pretty much work, and neutrophils. Except for B cells and neutrophils, I pretty much work on any cell, depending on, whatever we're interested in the lab. And so at the beginning, we know nothing, and then we develop various assays, and we actually help pushing the field a little bit more, I think, and then we make our little contributions. So I'd like to talk about two cell types, macrophages and then T cells. So macrophages, as I'm sure you know, you hear a lot from Sheila, are innate immune cells that are best known, and that's why they're called macrophage. They're best known for their capacity to engulf and destroy various kinds of things that the body doesn't want. So this is pathogens, dying or dead cells. This is a process called efferocytosis, stress cells and aging living cells, and that happens every day. We eliminate billions of cells through macrophages every day, and we need to do that. It's very quiet, very silent, doesn't cause any disease, and it's a key component of the normal immune function. Eliminate all these cells that are too old or starting to be a little bit dysfunctional. And of course, they can also eliminate tumor cells. And that's why people have been interested in targeting macrophages, perhaps as in the context of a cancer immunotherapy. And the main mechanism by which they do this engulfment is called phagocytosis. Okay, so what basically, the macrophage basically eats the entire target cell. They can also do other things, the macrophages, and I'm not going to talk about this today. And actually, we don't really study much of these things in our lab, but we should, because a lot of the pathways that I'm going to be talking about not only regulate phagocytosis, but they also regulate other types of macrophage functions, such as phagocytosis, which are basically macrophages nibbling little bits of living cells until the cell dies. Neutrophils also do that. Dendritic cells can do that, but macrophages do that as well. They can do cytotoxicity through various effectors. They can produce cytokines, and they can present antigen to T cells. So they have a lot of other functions in addition to phagocytosis. So this is a review we just wrote for Nature Reviews Cancer. So you can see here that the process of phagocytosis involves multiple steps. And when people talk about checkpoints that control the phagocytic process, it can actually be at any of these steps. So at the beginning, of course, you get a tumor cell or any type of target cell, could be a bacteria too, that will release some signals that will enable attraction of the macrophage towards the target cell. So therefore, you get chemotaxis. And then there's an important step here, which is conjugate formation. Which is actually controlled by charges. So negatively charged things like sialic acid, and I think probably chondroitin sulfate would be in there as well. They can actually enable repulsion between the tumor cell or the target cell and the macrophage. And this is still not really the phagocytic event itself. And the phagocytosis starts when you get extension of pseudopodia towards the target cell with formation of phagocytic cup. Then you get formation of a phagosome, fusion of the phagosome with lysosome, which basically leads to degradation of the target cell through various processes. And some of these things can lead to the generation of peptides that can then be presented by the T cell receptor to activate other types of immune cells, in particular, of course, T cells. And that's how T cells can actually be part of the effector mechanisms of macrophage activation. Now, as I said, there are a variety of receptors that control this process. There are some what we call eat me signals, which are basically prophagocytic receptors that will actually, oh, I realized they screwed up my slides here. This is the journal that did that. Oh my God. Anyway, so there are some eat me signals that will trigger the phagocytosis. So they should be here in red. So there should be a preponderance of red here. And then there's some don't eat me signals, which are the blue ones, I guess. I don't know, maybe it's okay. Anyway, whatever. So if you get more stimulation of prophagocytic receptors, you get phagocytosis. If you get more stimulation of inhibitory receptors, then you prevent phagocytosis. So same principle as the other types of immune cells. Now, this is a summary of the likely or well-demonstrated prophagocytic and anti-phagocytic receptors. So those eat me and don't eat me receptors on the macrophage. I think Fc receptors for immunoglobulins is the most predominant one, but we published a number of years ago that a member of the SLAM family receptors, SLAM F7, is also involved in recognizing ligands on tumor cells, for example, to trigger phagocytosis. The integrin, CD11A, B, C, in combination with CD18, LRP1, some galectin. So there's a variety of different types of prophagocytic receptors that exist on macrophage that can be involved in phagocytosis of target cells, including cancer cells. By opposition, there are anti-phagocytic receptors as well that balance this, and they provide the don't eat me signals. The best one is probably SIRP-alpha, which recognizes CD47 that is broadly expressed on normal cells, often overexpressed on cancer cells. But there are other things as well, like PD1, Siglec-10, LILRB1, 2B4, LY9, and I'll talk a little bit about this one, CD200R1 as well. So there's these variety of receptors that macrophages have that recognize ligands that may or may not be present on potential target cells, including tumor cells, and this balance will determine whether a target cell is phagocytosed or not by the macrophage. So we've been interested for a number of years now on SIRP-alpha and CD47, which is one of those inhibitory checkpoint. So SIRP-alpha is an inhibitory receptor, so an anti-phagocytic receptor that recognizes CD47 as a ligand that is expressed on and SIRP-alpha is expressed on macrophage, DCs, neutrophils, NK cells, and neuron. In contrast, CD47, the ligand, is broadly expressed on all normal cells. So it's a bit of a problem targeting this pathway because the ligand is expressed everywhere. So if you try to block this, you risk having also increased phagocytosis of normal cells, which can, of course, lead to problem in the clinic. But what people argued was indication that this may be a good target against cancer is that CD47 is often overexpressed on cancer cells. And actually, I think this is something that's probably true for the majority, if not all, cancer types. Now, Irv Weissman and also Jane Danska in Toronto, but Irv Weissman at Stanford initially proposed that if we could block the SIRP-alpha CD47 immune checkpoint with blocking antibodies, we could enhance phagocytosis and we could perhaps also help treat cancers when there are macrophages in the tumor. So there have been some phase 1 and 2 studies of SIRP-alpha CD47 blockade, plus usually tumor opsonizing antibodies to stimulate Fc receptors against a variety of cancers. And these have been quite promising, especially leukemias and lymphomas, and also Sézary syndrome, which is a cutaneous T-cell lymphoma. So this led to a lot of excitement in the field that we had identified an innate immune cell checkpoint that could be a target for therapy. And a lot of companies invested a lot of money in this approach all over the world. And it unfortunately was a disaster. And what happened is people realized that blockade of CD47, in particular with this antibody developed by Irv Weissman, magrolimab, led to anemia, thrombocytopenia, when it was given with CAR T-cell lymphopenia. There were some clinical studies where people died. where tumors got worse, got bigger. And as a result, some clinical trials had to be terminated. And you know how the pharmas work, right? And biotechs work. If there's a bad news, then they all run away, right? So as a result, this potentially very interesting approach of blocking this inhibitory immune checkpoint led to a much decrease in enthusiasm. And of course, it's not completely dead. I think there's still groups doing this, and there's still some interesting results that are coming out, especially with agents that like SIRP-alpha CD40, SIRP-alpha Fc fusion protein that binds and blocks CD47. But obviously, one question we've asked in the lab, because we're interested in mechanism, is can this approach, which looked great but had recent problems, can that be improved so that we can perhaps still get good results with this novel approach? So we've been interested in figuring out how SIRP-alpha works. As I pointed out here, one of the major mechanisms of action of SIRP-alpha expressed on macrophage, and it's an inhibitory receptor, is that it recognizes CD47, and that will transduce inhibitory signals that prevent phagocytosis. But this was not as efficient as one would like. And we were interested by the possibility that there may be some additional mechanisms of action of SIRP-alpha that we don't know about, and that these mechanisms of action also should be considered if we try to develop therapeutics that will target the SIRP-alpha CD47 immune checkpoints. So here what we have is a variety of different macrophages. So these are bone marrow-derived mouse macrophages. So normal macrophage express SIRP-alpha, and here you have tumor cells that express CD47. So here you get wild-type molecules of SIRP-alpha that bind to CD47. We also have SIRP-alpha knockout macrophage, which have lost SIRP-alpha. We generate those by knockout strategy. And then we put back a variety of mutants of SIRP-alpha that can or not bind CD47. And we're trying to see if you eliminate CD47 binding, does SIRP-alpha still have some activity? And maybe this activity also should be targeted when we're trying to block the SIRP-alpha CD47 checkpoint. So the first mutant that we put back in those cells, there's a mutant in which all the tyrosines in the cytoplasmic domain of SIRP-alpha, and there are 4 of them, which recruit the inhibitory effectors of SIRP-alpha, are mutated to phenylalanine. So this should be a signaling defective mutant. We get the same thing also if we basically delete the entire cytoplasmic domain of SIRP-alpha. We also created a mutant in which the binding site on SIRP-alpha for CD47, and this is this 3-adenine 95V here, is mutated to a valine. And you can, or is it 96 or 95? 95 or 96, whatever, I forgot. So we make this mutant, that this mutant, the SIRP-alpha, should still be able to get phosphorylated in the cytoplasmic domain, but should not bind to a CD47. Then we have the double mutant in which we get the CD47 binding site mutation, that is here. And then we get on top of that the phenylalanine substitutions in the cytoplasmic domain, and the same thing with the cytoplasmic domain truncation in addition to the it's actually 96V mutation. So then we have all these different macrophages that express all these SIRP-alpha mutants, including some that don't have cytoplasmic domain, some that don't bind to CD47, and the combination. And we look at whether they can or not suppress phagocytosis, okay? So this is the wild-type macrophages, and this is the extent of phagocytosis that we measured with a Frodo red-based assay, so you get 10%. And this is when you don't have SIRP-alpha, it's about 20 to 25. Now, if we put back the wild-type SIRP-alpha, you can basically suppress again the phagocytosis. So this is, of course, the wild-type molecule. It's working well. And these are all the various mutants. So you have a mutant here that doesn't have the cytoplasmic domain tyrosine. This one doesn't have the cytoplasmic domain sequence in its entirety. This is the mutant that doesn't bind CD47, and these are the double mutants that have lost both function. And you can see that none of these mutants is able to suppress phagocytosis to the same extent as the wild-type molecule. So even though they never are as effective as, you know, as defective as the mutants or the cells that are lacking SIRP-alpha. So therefore, there's something independent of CD47. There's something independent of these tyrosines that enable SIRP-alpha to mediate some inhibitory effects, right? And you get here a quantification from multiple experiments. So what is this? So we spent a lot of time working on this. Sorry, here my alpha got replaced by little boxes. That's probably a conversion thing with the stick. So we spent a lot of time trying to figure this out. And what we figured out at the end is SIRP alpha, in addition to recognizing CD47 on tumor cells in trans, it also has a cis binding mechanism, whereas it interacts in cis with CD18, which is a component of the CD11 integrins. And that's an additional mechanism by which SIRP alpha suppresses phagocytosis. So what we did is we took either wild-type macrophage or SIRP alpha knockout macrophage, we lysed them, and then we immunoprecipitated SIRP alpha, and we tried to identify proteins that were co-precipitating with SIRP alpha. And this is what we came up with. We found CD45, a very abundant phosphatase on all immune cells. It's not important for this effect because we did genetic studies, and lacking CD47 does not impact the residual inhibitory function of SIRP alpha. But we also found CD18 and CD11B, which are components of MAC1, which is one of the receptors also involved in phagocytosis. Now, this is an example of evidence that indeed, not only these two proteins co-precipitate in mass spec experiments, but also they're actually in very close proximity. This is a FRET-based assay where we give one color to SIRP alpha, one color to CD18, and then we look for evidence of proximity. And you can detect proximity by basically bleaching the acceptor, CD18. And if CD18 is close to SIRP alpha, what will happen is that the fluorescence of SIRP alpha will go up after the bleaching. And so you basically, if it goes up from here to here, it means that there was physical proximity between the molecule. So there's actually a good FRET, a good energy transfer between SIRP alpha and CD18. It's blocked by an anti-CD18 antibody. It's not blocked by an anti-CD11B antibody, which we also transfect in the cells, although there's no tag there, indicating that probably SIRP alpha is directly binding to CD18, and then CD18 is binding to CD11B. So we tried to figure out exactly how SIRP alpha was binding to CD18 and cis, and see if it was the same sequences that were involved in the trans binding to CD47. So I told you there's a mutant of SIRP alpha, which is the threonine 96V, that cannot bind to CD47. And what we were hoping is to identify mutants of SIRP alpha that can bind to CD47 but cannot bind to CD18, right? To show that actually the binding sites are different. So to help doing this, we took advantage of the fact that SIRP alpha has some cousins or some brothers and sisters, and one of them is SIRP beta, which doesn't bind CD47. And you can see also here, it doesn't bind in cis to CD18. And then we started to make chimeras between SIRP alpha and SIRP beta to try to see, okay, what is responsible for SIRP beta not binding CD18, and try to bring that in SIRP alpha and now make a SIRP alpha mutant that cannot bind CD18. So we identified a bunch of residues after doing all of this that were critical for that. And there's one mutant in particular, R91T, which is different from the T96V, which is required for binding to CD47. And this R91T mutant can no longer bind to CD18. So therefore, it's different residues, although they're very close, but different residues are involved in the cis interaction between SIRP alpha and CD18 compared to the trans interaction between SIRP alpha and CD47. Now, this R91T mutant that cannot bind of SIRP alpha that cannot bind CD18 is, however, capable of binding very well to CD47, that's shown here. So really, the binding sites for these two partners, whether it's the cis binding to CD18 or the trans binding to CD47, are different. And I think if one wants to block this pathway with antibodies, you can target CD47, and that's great, but you're still going to get the CD47 independent function of SIRP alpha, right? And if you target SIRP alpha, you have to be careful. You have to have antibodies that will block both binding sites. Otherwise, you'll just have a partial blocking of SIRP alpha. So to show that this dual mechanism of action of SIRP alpha is important for phagocytosis and control of phagocytosis and control of anti-tumor immunity, we tried to generate antibodies that were defective in one or both binding sites in SIRP alpha and see if you get the best suppression of the function of SIRP alpha when you block both of the binding mechanisms, right? So here we have the same experiment as before. So we have our knockout SIRP alpha deficient cells in which we put back either a wild type SIRP-alpha, as shown here, R91T, which is a mutant that cannot bind to CD18 but can bind to CD47. And then we have the mutant that cannot bind to CD47 here. That's the T96V, but can bind to CD18. And then we get the mutant with the phenylalanine, and we get the double mutant. So this is an important one here, the double mutant, in that we have basically this mutant. It has both the inability to bind CD18 and the inability to bind CD47. So that's the one we would think would be the most effective in the ability to suppress phagocytosis. So these are phagocytosis assays. This is wild-type macrophage, SIRP-alpha knockout macrophage. There's an increase in phagocytosis, about 2.5-fold. We put back wild-type SIRP-alpha, so we suppress completely phagocytosis to the same as the wild-type cells. These are the individual point mutants. So here we have the R91T. So this one is defective in binding to CD18. So you see it still suppresses phagocytosis, but not the same extent as the wild-type SIRP-alpha. This is the single mutant that cannot bind to CD47. Again, it partially suppresses phagocytosis. And this is the double mutant here. So you can see that when you mutate both sides, both the ability of SIRP-alpha to bind CD18 and the ability of SIRP-alpha to bind CD47, this is when you get the most compromise in the ability of SIRP-alpha to suppress phagocytosis. And it's the same effect as if you had no SIRP-alpha. So therefore, really, if you want to eliminate fully the function of SIRP-alpha, don't focus only on CD47. Remember that there's an additional mechanism of action of SIRP-alpha, which looks like it's responsible for 50% of the inhibitory activity, which involves a cis binding to CD18, an integrin that is critical for phagocytosis. So is this important in vivo, or in mice at least, in the ability of SIRP-alpha to suppress phagocytosis of tumor cells and elimination of tumor cells? So here we wanted to create antibodies that can block either binding of SIRP-alpha to CD47, binding of SIRP-alpha to CD18, or both, okay? So the way we did this is by creating bispecific antibodies. And this is some work that we've done with actually support by BioCanRx. So you can see here that we have a variety of mouse SIRP-alpha antibodies that we've generated in the lab. And we looked at the ability of these antibodies by FRET assays to suppress either the SIRP-alpha CD47 interaction or the SIRP-alpha CD18 interaction, okay? And you can see here in the FRET assays that we have an antibody, clone 17, that is very good at suppressing the cis interaction between SIRP-alpha and CD18. So we've got one that targets very well one interaction. Other antibodies have little or no effect. And here, when we look at the effect of these antibodies on binding to a CD47 Fc fusion, so this is a trans binding to CD47, clone 17 is not very good, but we've got two antibodies that are very good at blocking this trans interaction. So it's not perfect. So there's some effects of, for example, clone 17 on the trans interaction, but we thought that if we put both effects together, so if we had a mix of clone 17 and a mix of clone 27, that would be how we can maximally affect both the SIRP-alpha CD18 interaction in cis and the SIRP-alpha CD47 interaction in trans. So we made these bispecific antibodies that had one arm that was clone 17, that's good at blocking the cis interaction with CD18, and one arm of clone 27, that's good at blocking the SIRP-alpha CD47 trans interaction. And then we look at the effect on phagocytosis, and we look at the effect on tumor growth. So if we have clone 27, which is good at blocking the trans interaction with CD47, you can see that you do increase phagocytosis, okay, as you would expect. And we've published that already. If you have clone 17, which is especially good at blocking the cis interaction, although it also blocks a little bit the trans interaction, you can see that you get a partial effect. You don't know which mechanism is involved. But if you have the dual capacity of the antibodies to block maximally SIRP-alpha CD18 and SIRP-alpha CD47, you see this is when you get the most increase in phagocytosis. And it's true for these two different targets. So this is like kind of, therefore, I guess, cell antibody-mediated evidence that you need to complement these two functions or block these two functions of SIRP-alpha to have the maximal effect on phagocytosis. And this is a tumor progression assay here where we inject tumors in mice, and then we look at what happens if you block SIRP-alpha with clone 27, which is good to block the trans interaction, clone 17, which is good to block the cis interaction, or the bispecific. So if you have the antibody that blocks the trans interaction with CD47, you suppress tumor growth, and that's great. If you have one that blocks the cis interaction, but has also a little bit the trans interaction, you get a partial effect. But really, if you have this bispecific antibody that is active, most active at both functions at targeting both functions of SIRP-alpha, this is when you get the most suppression of tumor growth. So therefore, we think that SIRP-alpha has a dual mechanism of action. It wasn't known before. So it has a CD47 dependent mechanism of action, which is the one described in all the textbook. And this is great. And I think it's true and it's real. And we've confirmed that. And this is something that's working via these tyrosines within the cytoplasmic domain that recruit effectors, probably SHP1, that suppress things like VAV1. So that's mechanism number one. We think it's responsible for 50% of the function. But we think there's another mechanism that involves a cis interaction of SIRP-alpha with MAC1 or CD11b CD18 via CD18, and that this interaction in cis suppresses the activation of MAC1 that is needed for phagocytosis. And this is, we think, another 50% of the mechanism of action of SIRP-alpha. And I think if you want to block completely the SIRP-alpha CD47 checkpoint, you should not only target CD45, but you should all target the CD47, but also target the ability of SIRP-alpha to bind to MAC1. So I think this is best achieved with some anti-SIRP-alpha antibodies that would target both interaction rather than anti-CD47 antibodies, which is what most people have used so far. So, and these antibodies should not only block the SIRP-alpha CD47 interaction, but also make sure that they block the SIRP-alpha MAC1 interaction. Now, we've tested a bunch of clinically tested antibodies against SIRP-alpha that people have used in various clinical trials. Of course, the anti-CD47 antibodies don't block the SIRP-alpha MAC1 interaction or SIRP-alpha CD18 interaction. I think that's partly why they're not as great as we would like. But the anti-SIRP-alpha antibodies, most of them, they only block the SIRP-alpha CD47 interaction. This is how they were developed. They were tested for that. But there's one antibody that actually we found, and it's in our paper that's published, Core 23, that actually blocks both interactions. So you would think this would be the best antibody to test in the clinic. Unfortunately, this antibody is great at blocking both functions of SIRP-alpha, but it also cross-reacts with SIRP-beta and SIRP-gamma, which are cousins and brothers and sisters of SIRP-alpha, and screw up, I think, everything. So you really need to have antibodies that will target both functions, but also you have to make sure they don't target the cousins and the brothers and the sisters of SIRP-alpha. Otherwise, you're going to get other effects in the immune system. So I think this is a good example of how a better understanding of how mechanisms of actions are. Things are usually more complicated than we think. You get these simple little cartoons, that you get in textbook, and sometimes they're the basis for how people do drug development. And I think things are usually more complicated. And I think knowledge of this complexity is important to develop the better drugs, to make sure or to test best if something is a good target for therapy or not a good target for therapy. So I would think that if you really wanted to develop, target this checkpoint and get the best assessment of whether it's a good checkpoint to target for cancer treatment, that you should target SIRP-alpha and you should target both functions of SIRP-alpha. So this dual mechanism of action of SIRP-alpha is actually quite interesting because we had reported the same thing, a dual mechanism of action also for CD47. So SIRP-alpha has a mechanism of action that's CD47 independent, but CD47 has a mechanism of action that is SIRP-alpha independent. And again, things are more complicated than we think. We think there's a single mechanism of action, and many things I think have multiple mechanisms of action. So CD47, for example, of course, part of its effect is because it binds to SIRP-alpha, as we just described, but we also reported that in cis, CD47 binds to SLAMF7, which is a ligand for the prophagocytic receptor SLAMF7. It's a homotypic receptor. So on tumor cells, CD47 binds in cis to that, and it's preventing SLAMF7 from engaging SLAMF7 on macrophages, which is responsible for phagocytosis. So again, CD47 also has a dual mechanism of action, at least dual. There may be other ones that we don't know about. And of course, both mechanisms would have to be targeted if we want to have effective therapies. So this is SIRP alpha CD47. I was discussing with Jason Moffat yesterday. I think for now, we're a little bit, we did everything we wanted to do with SIRP alpha. We're probably going to find other things to do. But I think that, I think what people need to do now is generate better therapeutic tools that target this pathway and do better clinical trials. And I'll come back in a second how I think this could be done. Now, another thing is that SIRP alpha is not the only inhibitory receptor on macrophages. There may be other ones that could be targeted either to use instead of SIRP alpha, if SIRP alpha CD47 blockade is too toxic, or maybe complement. There's more and more studies done where people use combinations of immune checkpoint blockers. And there was a lot of publicity around PD-1, which to me was like a overinflated publicity, Siglec-10, LILRB1, 2B4, we published that, so that's good. So why not? But one of the things that we've been interested in recently is another inhibitory receptor expressed on macrophage, CD200R1, that recognizes as ligand CD200. And we think this is also an interesting checkpoint, and it's a true inhibitor of phagocytosis. And we have some data that actually you could block not only SIRP alpha, but also CD200R1 together. And that may be also more effective than just blocking CD200 SIRP alpha alone. So CD200 is actually much more restricted in its expression pattern than CD47. This is a bunch of tumors here. You can see the more pink it is, the more the tumor types express CD47. You see there's a lot of pink everywhere here in these various clusters. But if you look at CD200, there's a lot of blue here. So a lot of tumors types do not express CD200. But there's some exception like CLL here or multiple myeloma here and some leukemias that actually have a lot of CD200. So there are tumor cells that express CD200. Obviously, if you want to target that checkpoint, you should use it against tumors that have CD200. So that would be important. But the good thing also about CD200 is unlike CD47, which is expressed on all normal immune cells. And that's a problem. That's why there's anemia. That's why there's thrombocytopenia. You can see that CD200 is actually much more restricted, and it's expressed basically on resting and activated B cells and activated CD4 cells. So it may be much less toxic. But does it work? Can you use it to target to improve phagocytosis and improve anti-tumor immunity? So we showed in this paper, it's published in Nature Communication about 6 months ago, you can block, this is basically, we can just focus here. This is basically a tumor growth here, and this is example with the luciferase reporter. So you can see that when you inject an antibody against CD200 here to block the CD200 interaction, you actually dramatically decrease tumor growth. And you see it here also over time. So we think that this is and this is through macrophages. So we think this is a bona fide suppressor of phagocytosis, the CD200, R1 CD200 inhibitory checkpoint. And if you block that when you have a CD200 positive tumors, it may also be useful to promote phagocytosis. And in this paper, we also showed that it's even better when you combine it with SIRP alpha CD47 blockade. So I think that's the other aspect that I think people have to think about when they block let's say SIRP alpha CD47, is that maybe it needs to be used in combination therapies, perhaps with by targeting other inhibitory checkpoints. So this, I've alluded to this before. So I think I would not give up on inhibiting phagocytic checkpoints for therapy against cancer. I think there's still a future out there. I think we just need to develop better understanding of how these molecules work, how we can target the function of these molecules, and then I think also do better clinical trials. So develop better drugs, of course, stemming from a greater understanding of how immune checkpoints and their drugs work. I think we rushed into a bunch of studies with magrolimab, for example, without really knowing how SIRP alpha CD47 works and how this antibody is functioning. I think we can also use combinations of anti-phagocytic checkpoint blockers, like we showed for CD200R1 and SIRP alpha. There could be other ones also that may be effective. I think you also, when you do therapies that target macrophages, and you want to increase phagocytosis, you should also make sure that you get enough stimulation of phagocytic receptor, prophagocytic receptor. I told you, it's a balance between the two. And if you just alleviate inhibitory phagocytic receptors, you're not going to get phagocytosis. You need to trigger some prophagocytic receptors if you want to get phagocytosis. This is usually achieved by opsonizing tumor cells with an antibody that engages the Fc receptors, but there are other targets, right, like SLAMF7, that also can be used to stimulate the prophagocytic receptors. Combination therapies with other modalities, not just different immune checkpoint blockers, but also perhaps chemotherapy, radiation therapy, and others. Use biomarkers, of course, to better identify patients that will respond to that. So far, there's been no biomarkers that people have tested to decide, okay, I should try this therapy for these patients versus these other patients. And lastly, one of the problems that we have now with all these trials that people have been doing and that they either stop or that they give up on or terminate because of toxicity is they involve tiny number of patients. And one trial of a drug developed in Toronto by Trillium was stopped because they could not find enough patients. So I think it's important also not just to focus on one type of cancer, but rather focus on maybe multiple types of cancer that maybe share common biology, and to do these what we call basket trials, right? Where you basically have multiple types of cancer that you test in relationship to a novel therapeutic modality. So second part, how are we doing with the time? 44, yeah, that's good. So second part of the talk, this is something new. So we used to work on T cells. I started my lab working on T cells. Then we, as I said, NK cells, the macrophage, now we're back with T cells. So T cells are very important, of course, for immunity and adaptive immunity, and they're important for anti-tumor immunity, but they're also involved in antiviral immunity, and they're involved in the pathogenesis of autoimmune disease. So they're really critical. And they can produce various cytokines and mediate target cell killing, and they can destroy infected cells, tumor cells. And like other immune cells, T cell activation is controlled by the balance in the degree of stimulation of various activating and inhibitory receptors. So the activating receptor, the major one, is the T cell receptor, but for antigen. But by itself, this is not enough. There are also co-activating receptors on T cells like CD4, CD8, CD28, and ICOS. And this is counterbalanced by various inhibitory receptors, such as PD-1 and CTLA-4. And these are molecules against which drugs have been developed to enhance T cell activation, and they're quite effective in the treatment of certain types of human cancer. Actually, more and more of them are identified, like melanoma, renal cell carcinoma, non-small cell lung cancer. But there's some tumor types that don't respond at all. But these are really this is the biggest advance, I think, in cancer therapy of the past 20 years. And people have gotten a Nobel Prize for this. Now, as I said, blocking these two inhibitory molecules, PD-1 and CTLA-4, is very effective against certain types of cancer, but not in all patients having these cancers and not in all cancers. So in some cancers, they don't work at all. And people have suggested that we need to keep on looking for new T cell inhibitory checkpoints that we could target for therapy. And people have looked at things like TIM3, TIGIT, LAG3, NKG2A, and the results have been very disappointing, very disappointing. I think almost universally disappointing, except for one situation with LAG3 antibodies in combination with anti-PD-L1 antibodies for colorectal carcinoma. So, but other than that, a lot of disappointments. So there comes our little attempt to develop new therapies against what we think is a now established inhibitory T cell checkpoint. So it's one of the SLAM family receptors that's called SLAMF6. Now, like other SLAM family receptors, SLAMF6 is a homotypic receptor, meaning that the ligand of SLAMF6 here on T cells is another molecule of SLAMF6. So it's a homotypic receptor. SLAMF7 is the same, except the ligand is SLAMF7. Now, SLAMF6 got a lot of publicity because it's selectively expressed on progenitors of exhausted T cells. So during cancer development or during viral infection, you get tumor T cells that become exhausted. They get very tired, right? They cannot function anymore. And one of the way you can distinguish these exhausted cells that are sort of hopeless from those progenitors that are still hopeful and can be rescued is expression of SLAMF6, at least in the mice. So these TPEX, the progenitor of exhausted T cells, they express high levels of SLAMF6, but the exhausted T cells, they lose it. So people got very interested. Oh, yeah, that must be an important molecule. But it's just a marker so far. Now, people have shown functionally all kinds of things about SLAMF6. Some people will say it's an activating receptor. Some people say it's an inhibitory receptor. And as a result of this, nobody's touching it as a potential therapeutic target. Nobody's touching it. So various SLAMF6 antibodies that have been developed, soluble SLAMF6 ectodomain. So this is a soluble fusion protein that basically has the extracellular domain of SLAMF6. Ectopic expression of SLAMF6 on tumor cells has been shown to modestly enhance activation of T cells and NK cells. So people said, there you go, it's an activating receptor, right? So leading to the idea that it's not maybe a good target for immunotherapy. People want to look for inhibitory receptors that you block. However, genetic studies in mice have shown that loss of SLAMF6 augmented activation of T cells. This is a paper published by Michal Lotem that we were co-author on. We published also a paper on NKT cells that SLAMF6 was also inhibitory there. That was in Nature Immunology, 2019. Also inhibitory in TFH cells, that was in JXMed, and also immature NK cells. And this is one of our last NK cell paper, and this is where SLAMF6 is involved in NK cell education by mediating inhibitory signals. So that's still, because it's a mess, people don't want to touch it with a 10-foot pole as a potential target for immunotherapy against cancer. So it led us to reassess whether SLAMF6 is activating, inhibitory, or both, and whether SLAMF6 could be targeted to improve anti-tumor immunity, when in situation where targeting of either PD-1 or CTLA-4 doesn't work. Oh, I hate these things out of focus. Anyway, so that's going to come out in a few weeks. And basically, the first thing, of course, we looked at is where is SLAMF6 expressed in T cells. So PD-1, which is shown here in comparison, you can see that it's not expressed in resting T cells, but it's induced in T cell activation, unactivated T cells. SLAMF6 in comparison is already there on resting T cells. All resting T cells have it. I think there's no T cell that doesn't have it, except the exhausted T cell, right, in the mouse, but all the other T cells have it. And it goes up with T cell activation. So that's a good expression pattern if you're thinking of something, you want to target the T cell to improve T cell activation. So is it activating or inhibitory? So of course we can do various types of genetic studies. So here we use SLAMF6 knockout mice. So here we show experiments with CD8 positive T cells that we stimulate here with an anti-CD3 antibody with or without an anti-CD28 antibody. So here there's no APCs, okay? If you stimulate with PMA-inomycin, the two T cell types respond the same way, but you can see if you incubate with anti-CD3 with or without anti-CD28, the SLAMF6 dependent T cells are responding more in the way of proliferation, IL-2 production, or interferon gamma production. So it does look like it's inhibitory. Same thing here if you use an antigen-specific system with APCs. So here we have the OT1 T cells, which are basically, it's a transgenic T cell receptor expressing CD8 positive T cells that respond to the OVA peptide, synfecal peptide. And then we basically take these T cells and stimulate them either, so the T cells are either wild type or knockout, and we stimulate them with APCs in the presence of peptide, the antigenic peptide, that are either wild type or knockout. So here we have 4 different combinations: wild type, knockout, knockout, wild type, knockout, knockout, okay? So you can see that when you lack SLAMF6 on the T cells, there's increased response, even when there's SLAMF6 on the APCs. If you don't have SLAMF6 on the APCs, the response are normal. So the SLAMF6 has to be on the T cells. And if you have the double knockout with the knockout of SLAMF6 on T cells and knockout of SLAMF6 on APCs, it's the same as if you don't have SLAMF6 on the T cells. So really what matters is the SLAMF6 on the T cells, and that's logical because here there's no PCs. The only cells having SLAMF6 here are the T cells. So SLAMF6 is doing something alone on the T cells to suppress T cell activation. We got the same result with CD4 T cells, and we did all kinds of different types of stimulation, and basically you need to have SLAMF6 on T cells. When you remove it, you get increased T cell activation. This is with the mouse. We also did that with human T cells. So here we took 3 different single guide RNAs against SLAMF6, and we did knockout of SLAMF6 on the human T cells. Now, all human T cells express SLAMF6, and these are the knockout cells. And then we stimulated them here with anti-CD3 antibodies, and you can see that compared to the wild-type T cells, the SLAMF6 deficient human T cells have enhanced T cell activation response, including proliferation, IL-2 production, and interferon gamma production. And they also have an increase in TCR-induced protein tyrosine phosphorylation. So what SLAMF6 is doing is to actually suppress proximal signaling from the T cell receptor just by being there. SLAMF6 is there and it's suppressing T cell receptor signaling and the most proximal signals. And this is the same thing in the mouse. Now, we figured out here with some siRNAs, and we did the same thing also with CRISPR-Cas knockout of SHP1, that the actual effector of SLAMF6 that inhibits is this phosphatase SHP1. So, therefore, we had these results. So it's clear that SLAMF6 is inhibitory. And what we thought is that when people used antibodies against SLAMF6 that were enhancing T cell activation, maybe they were using blocking antibodies that were blocking the inhibitory function of SLAMF6, therefore resulting in enhanced T cell activation. But the antibodies are not that great. They increase activation by like twofold. So we wanted to develop better anti-SLAMF6 antibodies. By the way, that's why I think we got this paper accepted in that journal. So the current situation, as I said, is the available human SLAMF6 antibodies like NT7, 1B3, and we made a bunch of antibodies that people have described patents on. You know, you just get the sequence and you make them yourself, right? So they all cause a very modest increase in T cell activation. So we decided to make our own collection of novel human SLAMF6 monoclonal antibodies and elucidate the impact on SLAMF6, their impact on T cell function, their impact on anti-tumor immunity, and the goal is to develop antibodies that, completely block the function of SLAMF6 and recapitulate the phenotype of SLAMF6 deficiency. Okay. So we made all these antibodies and we did a bunch of assays. So this is, of course, T cell activation assays. So these are actually mouse T cells that express human SLAMF6. So we take SLAMF6 knockout mouse T cells and we put back human SLAMF6 there because these antibodies cross-react with human SLAMF6, right? And they don't cross-react with mouse. So you can see that when you get this antibody that's published already from you can buy it actually, NT7, you get a tiny increase in T cell activation response, right? So this is like this purple thing. So you get a tiny increase in T cell activation response to anti-CD3. Here, tiny, okay? So this is the best that's out there. Now, these are our antibodies, 5 of them, 5, 11, 21, 22, 23, and you can see that they have various effects on T cell activation, but we've got these 2 big winners here. Blue, which is number 21, fantastic at enhancing T cell activation, and 23, which is not as good, but also very good. Other ones, 22, 11 have no effect, and 5 is a partial stimulator of T cell activation, right? So how does it work? These cells only have SLAMF6. There's no APCs. So what are the antibodies doing? And we went back to our cis thing. So you remember SLAMF6 is a homotypic receptor. The ligand of SLAMF6 is SLAMF6. So we thought that what SLAMF6 is doing is being triggered in cis on these T cells, side by side. One molecule triggers another one. And that's mediating inhibitory signals. And what the antibodies are doing, we hope, is to block this cis interaction to alleviate the inhibitory effect of SLAMF6, promote T cell receptor signaling, and promote T cell effector functions and anti-tumor immunity. So we go back to our FRET assays here, and we basically put back one SLAMF6 molecule, this is human, one SLAMF6 molecule that's green, another one that's red, and then we do the FRET assays. And as I told you, what we're looking for is an increase in green between here and here, which is when you do the photobleaching, okay? So you do get energy transfer between the 2 SLAMF6 molecules. There's no effect if you have a control IgG. If you had this crummy commercial antibodies and T7, you get a little bit of a decrease in the FRET assays, so it's blocking a little bit the cis interaction. And then if you look at the other one, the best at blocking the cis interaction is this clone 21. You remember that was the one also that was the best at enhancing T cell activation. And the second best is this clone 23, which was the number 2 best. And then you've got the other ones. So there's a correlation basically between the ability of SLAMF6 to inhibit T cell activation and the ability of the antibodies to enhance T cell activation. So the more an antibody can block the interaction, the more efficient it is at enhancing T cell activation. And as I said, the winners is this clone number 21. So does this have an effect in vivo? So here we did various models. We did EG7, which is a lymphoma. We did B16 melanoma, MC38, which is a colorectal carcinoma. And we injected those in mice. Then we put back T cells that express human SLAMF6 instead of mouse SLAMF6. And then we treated them with these various SLAMF6 monoclonal antibodies. And we did some experiment. We also used anti-PD-L1 antibodies to see if there was synergy. So you can see if you have just the T cells by themselves without antibodies or with the control antibodies, basically the tumors are going to grow. If you put clone 11, which was non-blocking, the tumors are still going to grow. But if you put clone 21, which are the best antibody at blocking, you can see that now the tumors will basically disappear. You get the same thing if you put SLAMF6 knockout T cells. So really, this is phenocopying what happens with SLAMF6 deficiency. There's an increase in the number of T cells that produce TNF-alpha and interferon gamma. That's great. And then if you look at exhaustion marker here, including expression of TIM3, which goes up in exhausted T cells, and TCF1, which is lost on exhausted T cells, you can see that clone 21 here is great at rescuing expression of TCF1 and great as suppressing expression of TIM3. So we think that these antibodies targeting of SLAMF6 is actually, they had us rewrite this sentence several times. But basically, if I say it in court, it's basically reducing the proportions of exhausted T cells. So I cannot say it's preventing T cell exhaustion. It's just reducing the proportions of exhausted T cells, whether it's because it's expanding the other ones, or whether it's because it's getting rid of exhausted T cells, or it's reverting exhausted T cells, I don't know, okay? But it's reducing the proportions of exhausted T cells. I think we don't know better, by the way, even for the other ones, PD-1, PD-L1. I don't show it here, but if you do the combination of SLAMF6 blockade with PD-L1 blockade, it's even more efficient than SLAMF6 alone. So therefore, what we found is the homotypic receptor SLAMF6 suppresses T cell activation in response to a variety of stimuli, even when there's no APC, even when there's no SLAMF6 on APCs. And this effect is mediated by the phosphatase HP1. SLAMF6 interacts in cis. And, these cis interactions are very, very, I think, probably we were talking about that at dinner yesterday. They're very, very prevalent. I mean, if a molecule is expressed in one cell and its ligand is expressed on the same cell, in all likelihood, unless they're in different compartment completely, they're interacting in cis, okay? And these molecules were synthesized possibly at the same place. They migrated from the cytoplasm to the surface together, and they've been sitting there together for a long time. And to really disrupt these cis interaction by some trans interaction, I think you have to work at it. That's why I think the trans interactions work best if the cell, expressing the receptor does not express the ligand, and it's fine. It's just waiting for the ligand to happen. But if the receptor and the ligand are co-expressed, I think in all likelihood, they're already binding, even at basal state. So we've shown that SLAMF6 interacts in cis with another SLAMF6 molecule at the T cell surface, that novel blocking SLAMF6 monoclonal antibodies enhance T cell activation and promote anti-tumor immunity. And this is independent of expression of SLAMF6 on tumor cells, because all the tumor cell models we've tested don't express SLAMF6. We don't know what would happen if they had SLAMF6. That's an interesting question. Future. SLAMF6 blockade reduces the proportions of exhausted T cells in various tumor models and all the tumor models that I showed you about. And we think that SLAMF6, who was sort of written off because people had all these data, it's inhibitory, it's activating. We think it's a pure inhibitory receptor, and it may indeed be a useful therapeutic target to promote anti-cancer immune response, and particularly for tumors that are not susceptible to PD-1 and CTLA-4 blockade, and it's independent of expression of SLAMF6 on the tumor cells. Now, of course, you have to worry, okay, are you going to induce autoimmune disease with this? Are you going to induce inflammation? Of course, this is something that we have to look for. We saw no evidence of, cytokine storm in the short-term assays that we did. And also you have to wonder, what about immune cells other than T cells that express SLAMF6? That's why we're going back to NK cells, because NK cells also express SLAMF6. So that's where we are. So we think SLAMF6 is an interesting receptor. I think it's a homotypic receptor. It's engaging itself in cis. This is suppressing tonically T-cell receptor signaling via the phosphatase SHIP1. And we've developed these great blocking antibodies that block this cis interaction and I think are great at enhancing T-cell activation and may have therapeutic value. So these are the people who did the work. Ming Chow is actually the guy who's making all our monoclonal antibodies. He's just like super rigorous and systematic, and that's what you need when you want to make monoclonal antibodies. So he's doing it with the hybridoma technology. Jin, I think, who was taking the picture, is not there, also does it, but by single cell B-cell RNA sequencing. Zengai did all the work on phagocytosis with SIRPalpha. He's now in Macau. He has his own lab in Macau. Jashin did the work on CD200R1. He's now in Cleveland. He's doing his postdoc. Binley did all the work on SLAMF6, and he will probably find a job in China. I think he's looking now. And that's it. This is the rest of the team. Thank you very much.
Thank you so much, Andre. That was amazing. I mean, I think you're being fairly modest about it, but you've actually discovered a new checkpoint.
I think we have.
Yeah. And the other thing that I think is really paradigm shifting for all of us working in this field is you've really uncovered the importance of cis interactions. And we were talking about this. It kind of opens your mind to start thinking about what new steric and biophysical interactions are happening at the cell surface that we're not even considering, because we're all thinking about the canonical, here's a cell with a ligand, here's a cell with a receptor, and here's how they interact. But I think this is another thing that we all are going to be considering now much more. So thank you. It's an amazing talk.
You know, I must say, so we're big into that now, and we have published several papers with different evidence. The guy who really started that or resuscitated that for the immunology is Enfu Hui, who did it for B7 and CD28 and all these things at UCSD. So I think, you know, obviously we were very inspired by his work and he collaborated with us, but lately we've been big boys and girls and we're doing it on our own.
Amazing. I'm going to open to questions for Dr. Veillette. Kim.
We're mentioning that they have been, that they tried to do soluble ectodomin, and I would have thought that would have been tried for the CD47 SIRPalpha rather. So if they're the homotypic, if you were to express this extracellular domain message, that would not promote more inhibition.
Now you're talking about which one?
So for SLAMF6.
SLAMF6, yeah. So that's an interesting story. So I think what their soluble ectodomain was doing was actually breaking the cis interactions, okay? So they made the soluble ectodomain, they got these results, and then they could never reproduce the results because they could never remake the same batch of soluble ectodomain. So it was impossible to figure out the mechanism. But I think the likely mechanism is that their soluble ectodomain was preventing the cis interaction. But I don't know because it's gone.
Then with the soluble, you will be able to target both the cis
and you are right. So, and that has been done, not in the clinic even. So there's like 2 SIRPalpha FC fusions that have really moved fairly well along the line in clinical trials. One is evorpacept, which is from an American company, and they're even in phase 3 trials, and it looks pretty good, okay? And I think it's because, I wouldn't be surprised that it's because it's hitting both mechanisms, as you just said. And there's also a product from China, IMN01, which also they're going to report. I mean, they're making a big deal out of it. So I don't know, you know, it doesn't mean good. Usually when things go down the tube, they don't make a big deal out of it. They're going to report the results in March. And I think you're probably right. So that these soluble FC fusions would, on the one hand, they would block the ability of CD47 to bind SIRP-alpha, right? But also they may displace CD18 from SIRP-alpha, right? They may compete. So we will see. We will see. But I think we'll see. I think that field is not dead. I think that therapeutic approach is not dead. It just started with the wrong way. People were using these anti-CD47 monoclonal antibodies, and they were just targeting only one component of the function of SIRP-alpha. And also, I didn't get into this, but the magrolimab is a human IgG4. So, you know, people used to say, oh, human IgG4 doesn't bind Fc receptors, so therefore you don't get any induction of, you know, macrophage activation because of that. So, but it's not true. So human IgG4 does bind Fc receptors. So everybody knows that now. So it was not a good idea. So people now are making Fc inert versions of these antibodies. All the antibodies we use are Fc inert. We mutate sites so that they don't bind Fc receptors. Sometimes you want Fc receptor engagement, like Herceptin, you need Fc receptor engagement. Rituximab, you need Fc receptor engagement to get an effect. But sometimes you don't want it because the effects are involved in toxicity. And I think magrolimab, this anti-CD47, was engaging Fc receptor. There's a compound from Toronto, Trillium, Ontorpacept, or TTI-621. It also binds Fc receptors. And I think it's probably also causing a fair amount of toxicity. Now they moved on to an Fc inert version, but unfortunately they can't find the patients anymore. So, you know, I think all of this is these trials where people rushed into these trials with incomplete knowledge, saying that IgG4 doesn't bind Fc receptor. That's false. And it's too bad. I hope it doesn't kill the approach. I think it's going to be resuscitated. But it could have led to killing this approach, although there was potential, and there is still potential.
Wonderful. All right, I think, oh, Ali, we'll have one last quick question.
I exhausted you with all my stuff.
How that relates to exhaustion, because with the data, it looked like you're increasing the sensitivity of the TCR simulation. So I was wondering how does that lead to a reduction in the amount of exhausted cells, or however you want to frame it, where you actually maybe see more simulation of the TCR, which may actually drive exhaustion. So I'm just wondering how your thoughts on that.
Yes. So I think this is true for any of these things, right? So even anti-PD-1 and all this. So we enhance T cell activation, but then, you know, and there was a paper in Nature about this recently. So you enhance T cell activation with PD-1 blockade, but then these exhausted T cells may not become memory T cells. They may just die, right? So you may be shooting yourself in the foot. So I think this is a valid concern. We're going to have to wait and see about that. Now, I don't think it's rescuing exhausted T cells. Now, I'll tell you. So loss of SLAMF6 in the mouse is seen in exhausted T cells, but in human exhausted T cells, and we have that in the paper. One of the reviewers probably knew this and made us write it in the paper. But if we've done also a bunch of analyses of published single cell RNA sequencing database, human exhausted T cells still have SLAMF6. So it's possible you're rescuing them, although I doubt it. I think once T cells are exhausted, they're basically not rescuable, but maybe they are. Maybe there's a subset they are. But the rest, I think probably what we see in our assays is because we're enhancing activation of naive cells, we're enhancing activation of effector cells, memory cells, and those progenitors of exhausted cells. I think that's why the exhausted T cells get diluted out. So it's the proportions that change. And I think they were right. I mean, we don't know, right, whether it's rescuing. I doubt it's rescuing. I think these exhausted cells are done.
Okay. Everyone help me thank Dr. Veillette for a wonderful talk.